GPS is armed. Falcon 9 is startup and is now controlling. Falcon 9 is configured for flight.
Go for launch.
Apex, this is resilient. Roger, go.
Crew 1 for all. Eman, 15 seconds.
Go for launch.
10, 9, 8, 7, 6, 5, 4, 3, 2, 1, 0. Ignition.
Go for launch.
Hello everybody and welcome to the FameLab International Final 2021. It is the one we have been waiting for, the big one, and I am so excited. Hello, I am your host. My name is Greg Foote. I am a science journalist and a producer. I make and host content on TV, on radio, on podcast and YouTube. I'm also a science communication trainer as well, and it is an absolute pleasure to be here. I am live right now in my spare room. Very welcome. The live chat is open, so do come and say hello. Talking of which, loads of you have been saying hello already in the live chat, so let's do some quick shout-outs. Who have we got? We've got Bank in Thailand. We've got Ivelo, it says greetings from Bulgaria and best of luck to all of the finalists. We've got, who else we've got? Sipiso watching in South Africa. The FameLab Ireland team are watching and saying hello on Twitter, saying they can't wait to see the performances. Full hello from Portugal. Anantha from Austria, Relitsa from Bulgaria, La Rose from Canada, Marco in Italy, one of last year's participants, in fact. Hello, watching. Good to have you back. Brenda in South Africa says it's rooting for FameLab Team South Africa. Gosh, so many of you watching. Hello, you are very, very welcome. Do keep those shout-outs coming. If you're watching this, I expect you know what FameLab is, but if not, here is a quick 2-minute introduction. FameLab is the leading science communication competition and training program in the world. Each year, thousands of researchers from across the globe receive training and then compete in heats. FameLab was created by Cheltenham Science Festival and is delivered globally in partnership with the British Council, making science accessible for all. FameLabbers have 3 minutes to capture their audience. 3 judges judge them on the 3 C's of FameLab: content, clarity, and charisma. And you, the audience, vote for your favourite 2. Each country crowns their national champion, and those national champions come together to compete for the title of FameLab International Champion. FameLab participants have gone on to present TV shows, give TED Talks, publish books, and so much more. FameLab is all about giving skills, confidence, and opportunities to scientists, engineers, and mathematicians to communicate their current research with the people that it impacts. So, this year, 23 countries took part in FameLab. In each of those, a final was held to crown a national champion. Those 23 national champions came together to compete in the FameLab International semi-finals. They were so good, I was watching. And 10 of those then made it through to this, the FameLab International Final 2021. And here they are. Here are all of our finalists. 10 of the world's most charismatic and chatty scientists and engineers. Lovely to see you all. So, yeah, tonight they're going to be battling for the prestigious title of FameLab International Champion, although it is a friendly battle, I'd love to say. As you saw in that 3-minute little introduction there, each finalist has 3 minutes to share their science in an engaging and entertaining way. And by the way, if the timer runs out, then we do let them finish their sentence or their idea. We're not that cruel. They can use props, but they can't use slides. And they'll be watched by 3 judges who will listen to their talk. They'll ask them a follow-up question each, and then, after all the finalists have presented, the judges go away and huddle and debate and argue and cry, whatever it takes, to choose the person who will be crowned the FameLab International Champion 2021. That champion will walk away with a prize, and this beautiful trophy, here it is, so shiny. And as wonderful as this is to watch for you watching, don't you think you just get to sit back and enjoy the show? Oh no, you have a job to do as well, if you wish, because you get to choose who to crown the FameLab International 2021 audience favourite. That person will then know forever that they were your champion, and they'll get a trophy too. That one that I've got over there. By the way, your audience favourite can be the same person as the judge's chosen champion or the judge's chosen runner-up, right? The decisions are totally independent of each other. How are you going to vote? Well, you're going to go here, and the audience vote is going to open after all finalists have performed. So I suggest that you jot down the name of your favourite speaker or speakers as we go along, so you know who to vote for. Details about how to vote, you can go and check them out. And as I say, we won't open them until all finalists perform. So now you know how it works. But before we get started, there is one more thing. Technology, as we know, often likes to not play nice, to cause a bit of a hiccup just when you least want it. So we have actually pre-recorded our 10 finalists' presentations. It just felt to us like the fairest way to do it, so we didn't fall foul of the bandwidth gremlins during the live show. So last week, our 10 finalists and those 3 judges jumped on a big international Zoom call, and they each took it in turns to present for 3 minutes and then take questions from the judges. However, that's as far as we went. The judges went away and deliberated after the final presentation. They made their decisions about the runners-up and the champion, but they did not tell the finalists, or in fact me either. So tonight, you're going to get to watch those 10 presentations, hopefully without internet wobbles. And then after you've seen the talks and the questions, we will get to find out together who wins. Plus there is, of course, the audience vote as well. That will be happening live. As I say, we'll open the vote after you've seen all finalists' performance. And while you're casting your votes, to keep you entertained, I've got a few things for you. The first is a celebration of the 15-year partnership between Cheltenham Festivals and the British Council, and a look at what FameLab International has achieved with a special guest musical performance. And there's another one as well. It's the second thing. I'm going to have a live chat with an honoured guest who many of you may recognise. It's last year's winner. Think of all that, I guess, as the Super Bowl halftime show. Yeah, right. Enough admin, enough rambling from me. I think we'll all agree. It's time to get this show on the road. Let's meet the 3 people who will decide our finalists' fate. Here are our 3 judges and what they're looking for. See, it's hard to talk. What they're looking for in our finalists' presentations. Here they are.
Thank you. So my name is Dr. Tara Shine. I'm an environmental scientist. I'm based in Kinsale, which is on the south coast of Ireland, where I run a company called Change by Degrees that helps organisations to be more sustainable. And a key part of what we do there is helping them to better communicate sustainability. I'm also an author of a book about what everyday people can do to be more sustainable. And I work alongside and within the Climate Change Convention. So I'm just back from COP26. Nice to meet you all.
Hello everyone, my name is Julia Wheeler and I'm a writer, a journalist and a chairperson at the Cheltenham Science Festival. My background is very much journalism. I was with the BBC for most of my career, including as a Middle East correspondent for a long time. I am not a scientist. I read history, specifically economic and social history at university. So, and I haven't studied science since I was 16. So I'm really looking forward to learning so much while we're all together today.
Hello everyone, I'm Rogério Martins, I'm from Portugal. I'm a mathematician. I used to be a researcher, even if right now I do science communication most of my time. I was also a FameLabber in 2010, And right now I have a TV show here in Portugal on mathematics. It's called, you can see it on YouTube. It's called in Portuguese, Isto é Matemática, or This is Math in English. And I also have some stage shows, and I do essentially science communication and essentially in math. Even if math is something that connects everything, so usually I'm talking about every kinds of stuff that relates to math in some way, and that's what I do. Hope to I will enjoy for sure your thoughts.
What I'm looking for today is a finalist who really keeps my attention from start to finish, sparks my curiosity and tells me something I didn't already know.
What I'm looking for today as a writer is a sort of narrative drive pushing through the presentation. So something again that keeps my interest, but also perhaps has a beginning, a middle and an end, and is really well rounded.
What I'm looking for is to see someone explain a really nice piece of science, and explain it like if I was 4 years old, and with real passion. That's what we expect from people that are communicating science.
Tara, Rogério, Julia, thank you so much for taking on the challenge of judging our fantastic finalists. Rather you than me. Great to see so many of you saying hello in the live chat from all over the world. Who have we got? We've got Scott cheering us on from the Sunshine Coast. We've got Claudia watching from Mexico. Twitter, we've got Shaima saying all set in Egypt for the best event of the year. Yeah. We've got, who else? Edgar watching from South Africa. Lyubov and a group of viewers in Bulgaria. So many of you. Keep the comments coming. Keep them coming as we go through the show. Maybe also let me know, is there a fact that blows your mind? Maybe there's a prop that one of our finalists uses that you absolutely love. And keep those shout outs coming for where you're watching from as well. Let's do it then, shall we? Shall we get started? Let's hear from our first finalist presentation. Now, tonight's order was chosen by random, and kicking us off is Jesús from Spain. Jesús has a PhD in genetics and is fascinated by how we can understand human diseases by studying our DNA. When he's not in the lab, you'll find him in the cinema or nose deep in a book. Jesús's 3 minutes starts when he does.
Almost 100 years ago, on a tiny little island in the states of Florida, lived a famous writer named Ernest Hemingway. Hemingway was a cat lover who had heard tales about mysterious sailor cats with so many toes. One day, a sailing captain told Hemingway the reason why 6-toed cats were common crew members on many ships. They were thought to be a good omen, a symbol of good luck, or simply better mouse catchers. The captain gave Hemingway a white 6-toed kitten as a present, which he named Snow White. Developing extra fingers or toes is a genetic anomaly called polydactyly. Polydactyly can be present not only in cats but also in other animals, like humans. The same way changing words in a tale or story might lead to different endings, specific mutations in the DNA change its letters and cause polydactyly. Hemingway raised Snow White to become a beautiful cat that lived with not 7 dwarfs but 6 toes on its paws. And Key West, that tiny little island, soon started to see more and more 6-toed cats. Was it sorcery or some sort of spell? Absolutely not. Snow White, it turned out that Snow White was actually a male kitten, and that spread the polydactyl mutation all over the island. But how did polydactyly spread so quickly? Animals have 2 copies of their genes, one coming from the mother, one coming from the father. Generally, when one is broken, we use the good one. However, polydactyly is dominant, which means that inheriting the mutation, the polydactyl mutation, from one of the 2 progenitors would be enough to develop the polydactyl phenotype, more fingers or toes. In order to try and identify the genetic cause of polydactyly in that island, researchers sequenced the DNA of Snow White's polydactyl descendants. They couldn't find any mutation previously associated with polydactyly. Instead, they discovered that all the polydactyl cats presented the same but brand new mutation. The mutation became known as the Hemingway mutation because the family tree of the cats could be traced all the way back to that white six-toed cat that a captain gave once to Hemingway. The Hemingway mutation in cats is equivalent to human mutations that cause polydactyly as well. They turn on a very important gene on additional regions of the embryonic hen or paw, developing extra fingers. Developing extra fingers is a gain of function. It's not that the gene is broken, it's that the mutation confers new properties to that gene. In the case of polydactyly, these new properties create hen differences that can have no clinical impact or impair hen function. This is not a fairy tale, and genetics helps us understand true stories. If not, ask our Snow White, who ate no apple nor was destined to disaster, but lived happily and polydactyly ever after.
Jesus, well done. I love a story, love a bit of Hemingway in there. I wonder, where are we with this research today? I mean, Hemingway was a while ago. Where are we today?
Yep. So in polydactyly, there are many or several different genes that can develop the phenotype. So the developing of extra fingers. There are several genes, although each of them is able to create the phenotype, the developing of the extra fingers. We now know that there is a regulatory element, so it's not a gene, it's something that controls the gene, that is 1 million base pairs away in the DNA. So this is really far away, and this is really new, that we know that something from really far away can control a gene. So in this case, the polydactyl mutation that is found in cats, it's quite helpful because it's equivalent to human mutations that can be developed as well. So this is very useful in order to diagnose people, because when we have polydactyly, when someone shows polydactyly, it can be only having polydactyly or be the consequence of something more syndromic, so having more of the symptoms. So it's very important in diagnosis to isolate, which is whether it is something isolated which can have severe or milder consequences, or something more general that we need to classify and treat properly.
Thank you so much, Jesus. My question is, you mentioned that for the cats there might have been an advantage in having 6 toes for catching mice. Is there an advantage to humans in polydactyly?
Well, humans having polydactyly can have the same advantage or the same disadvantage as someone having blue eyes. So it's a change, it's a variation, a new trait or a different trait from common or from the majority of the population, but giving an advantage or not is not like the main thing. Sometimes when it's really severe and it can impair like hand mobility, so people can have some difficulties. But I would say a mild polydactyl phenotype is not either an advantage or a disadvantage.
Yes, I do have also a question. Actually, I do have some cases of polydactyly in close families. But I was wondering how frequent it is in humans.
So I don't know exactly the frequency, but it's a rare, I wouldn't like to say disease, a rare trait. So because in genetics we normally tend to forget about the medical thing, so we focus on the term, and we sometimes say genetic disease because it's something genetic, but doesn't really mean that it's a disease. So I really think we should be careful with the nomenclature there. It's a rare trait, so it's not something frequent. But if we add up everyone that is having polydactyly, we have many polydactyl people. So it's infrequent, but we are a group, or they are a group.
Nice one, Jesus. Kicking us off, first finalist. We've watched this. We've watched the performance. So many great comments coming in. Lots of support. Joanne in South Africa says great talk, very interesting. Dr. Bo says great presentation. Nian in Australia says good luck to all contestants. Eman, who is also a finalist, we'll hear from later, says amazing, Jesus. Lots of the finalists supporting each other, which is lovely to see, of course. Cabaret says all the best to every participant. Catgenic says very entertaining, Jesus. And we've also got semi-final judge Andrew Steele watching too, wondering who is going to be this year's 3-minute Psychon World Champion. Fantastic. All right, well, in which case, let's crack on, shall we? A reminder, you are seeing pre-recorded presentations and judges' Q&As today, but we are all watching them live together, and we're going to be revealing the runners-up, the audience favourite, and the winner live later, and the live chat is open. On to our second finalist then. Our second finalist is Samantha from Australia. Samantha is a former arachnophobe turned spider advocate and venoms scientist. Based at the University of Queensland, she recently completed her PhD where she used spider venoms to create new drugs against Australian sheep parasites and neglected tropical diseases. Samantha's 3 minutes starts when I press this button, and she does.
Hands up! Who here is afraid of spiders? I'm a former arachnophobe turned venom scientist and spider advocate. Because working with beautiful Australian spiders like Betty the Golden Orb Weaver taught me that while spiders may look creepy, the real creepy crawlies are the blood-sucking parasites living inside us. Parasites are a huge problem for both human and veterinary medicine. Malaria kills over 450,000 people each year. And here in Australia, our sheep are being literally eaten alive by blood-sucking worms. They can drain sheep of as much as 10% of their blood every day. And the problem is that we can't treat these diseases effectively. The parasites have evolved drug resistance. So we need new drugs to protect ourselves. So I'm asking the question, what if we try fighting the creepies with the crawlies? Spider venoms evolved over millions of years to have thousands of unique compounds. Some of these are fast-acting, potent, and selective, which makes them a great starting point for drug discovery. My research aims to take these drug-like molecules from venoms and turn them into new antiparasitic drugs. But how do you go from venom to drug? You need 3 S's: spiders, sheep, and shit. I work on a blood-feeding gutworm of sheep called the barber's pole worm, and I use this as a model for parasitic diseases. But because it's a gutworm, its eggs come out in the feces, so it's my job to dig through buckets of sheep manure over and over and over to find the microscopic worm eggs. Once I have the eggs, I can add the venoms and find out which ones kill worms. Then I look for the active molecule and synthesize it, which allows me to study how it works and make sure that it's safe to use. For my research, I led the largest ever screen of venoms against parasites, testing over 250 venoms against blood flukes, hookworms, roundworms, and malaria. And I found spider venoms that can kill every parasite we tried. We even found a molecule in tarantula venom that paralyzes human worms at a lower dose than the best antiparasitic drug ever discovered. And best of all, ours had no side effects in mice, making it a great clinical candidate. So, spider venoms could be the next generation of antiparasitic drugs. We just have to brave the creepies to fight the crawlies.
Thank you so much, Samantha, for this wonderful talk. And I was wondering what kind of effects will have in our real life, say in the future, your kind of research, this research.
Well, the real goal of my research is to be able to develop new antiparasitic drugs, which is critically needed because, here in Australia, we're very fortunate, and much of the world, Europe, for example, we don't even think about parasitic worms as a major issue. But the reality is that over 1 billion people worldwide are infected with parasitic worms. And we desperately need new medicines to be able to treat many of these parasitic diseases because we have drug resistance or a lack of efficacy. So what we're doing is hopefully finding new molecules that will be able to overcome that drug resistance. And the really cool thing about the spider venoms is that they work via completely new pathways. So the spider venoms are helping us to find new drug targets for the parasites as well. So it's looking to be a really sort of long-term pipeline of drug discovery against the parasites.
So who said science was glamorous, Samantha? I wonder what changed your mind in terms of working with spiders and how you can change other people's minds to get more people into this field.
Well, one of my key goals of doing FameLab is to help show people how beautiful and amazing and actually cool spiders are. You know, the majority of them are actually not dangerous to people at all. For example, Betty the golden orb weaver is completely harmless to humans, and she's pretty happy hanging out here with me on my hand. And the way that I got over my arachnophobia was really through education, teaching myself about how awesome these spiders are, how they play a really important role in our ecosystem, and also exposure therapy. I was genuinely arachnophobic when I started my PhD, so I volunteered. I challenged myself to get over my fear by taking care of the spiders. Then I named them, so I had Beyonce the tarantula, Whitney Houston the funnel web, and once you name them, you get attached to them, and they're not so scary after all.
I have a very unserious question for you, Samantha. In all of that sifting through the sheep poo, what was the weirdest thing you found?
The sheep poo is probably one of, actually, funnily enough, my preferred poos to sift through because they just eat grass, so it doesn't smell so bad. It's been much worse when I actually work on other types of parasitic worms like human and dog, and you have to dig through those feces. Probably the weirdest stuff has been when I've been doing dissections to actually collect parasites from different animals, and I was dissecting some fish, and as we opened up the fish there was this giant crustacean in the mouth, and it turned out to be a tongue-replacing isopod. Now this is a crustacean that swims through the gills of the fish, grips onto the tongue, squeezes the tongue to death, and becomes the tongue of the fish. And as I was looking at these fish for the different parasites, every single fish I had these parasites in them, and sometimes multiple fighting to be the tongue of the fish. So, you know, in comparison, the spiders are lovely. The parasites really are the creepy crawlies here.
Thank you.
Lots of mixed responses on the live chat to the spiders. Lots of people absolutely loving the talk. Tara says, great talk, very clear and engaging. Jessica says that spider is stunning. Darby says, very cute spiders. And then let's go to the other end of the spectrum. Irene says, slightly creepy, but fascinating talk. Marika says, no more spider. So should have come with a warning, I guess, that one, but fascinating. Thank you so much, Samantha. If you've just joined us, hello, welcome to the FameLab International Final 2021. My name is Greg Foote. I am your host for the evening from my spare room. A reminder that you will get to vote for the audience vote winner after all of the finalists have performed. Our third finalist is about to take to the virtual stage. Our third finalist is Sirawit, or to call him by his nickname, Ice. Ice is a lecturer at the Prince of Songkhla University in the south of Thailand. He's got a PhD in life sciences from Imperial College and loves using computers to solve biological problems. Sirawit's 3 minutes starts when he does.
Can you still remember the moment that you can travel anywhere you want to cherish good memories with your friends or family? For some people, this has been a while. The COVID-19 is now back on the rise again, putting many of us in isolation, and researchers are in the race against time to find a way to fight against this deadly virus. But developing a drug or a vaccine would be impossible if we don't understand well the protein component of the virus, such as the one on the surface here, because this could be the key to explaining how the virus infects us and help researchers find a cure. To get the crystal clear detail about the protein structure, there's a traditional method called X-ray crystallography, which requires you to use advanced technique in biochemistry to turn this tiny protein into a crystal. Then you shoot an X-ray beam onto it. As the beam hits the crystal, it scatters out, creating a fascinating pattern on the detector plate. This pattern is then analyzed by a computer and converted into a three-dimensional model of the protein. Impressive, isn't it? But that's a big problem. This technique requires lots of time and effort, and most of the time, scientists fail to get the crystal. Importantly, knowing just one structure is not enough because the virus is mutating rapidly. Thousands of COVID variants have been identified worldwide, and we don't have time to study all of them. But can we predict how they look using computers? Here's how it works. Think of a protein as a building block of amino acids. Normally, when it mutates, a certain spot gets changed, but the rest stays pretty much the same. So, rather than shooting an X-ray beam onto every mutated crystal structure that we have, couldn't we just use the model that we already have as a template and computationally simulate a new mutation onto it? This could give you a predicted structure in an instance. In my PhD study, I used this concept to create a web application to help researchers predict and analyze mutated structures. So far, this application has helped thousands of researchers investigate proteins in many species, from human to animals, plants, or even the coronavirus. This might be just a small step, but it could be the first step to more groundbreaking research to come to help stop the spread of the disease that has killed over 5 million people worldwide. And I've seen so many people losing their friends, lovers, or their entire family to the pandemic. And that is absolutely heartbreaking for me. If you are the one whose life has been affected by the COVID-19 as well, please rest assured that you are not alone. Please remember this word. With the current technology and all the effort from people around the world, we will fight together. And one day, we all together will put an end to this pandemic. Thank you.
Thank you, Ice. Amazing. My question is around the predicting that you're doing around the mutations. So how many different mutations have you been able to model in this predictive way already, just around, say, the coronavirus?
Well, in my research, I did the calculation. I tried to find out how many mutations first. And do you know what? I detect about 7,000 mutations just only on the spike protein of the virus alone. And this is the topic that I'm investigating in. You know, in terms of the virus, they have lots of proteins, but this is the protein of our interest. And we were able to also model about like hundreds of mutations because we were trying to focus on the mutations that were detected in my home country in Thailand, because, you know, this is going to help us, you know, monitor the virus mutation and try to let us find out whether the mutation is harmful or whether it's okay. Thank you.
Hello, Ice. I found really amazing your props, and I was wondering how this part that you have this crystal and then the laser. Could you explain a bit more how it works and how did you come with this idea?
Absolutely. Well, you know, it comes with the technique which is called the X-ray crystallography. This is not the X-ray, this is not the protein crystal. To be honest, it's just like a regular crystal that I got it from an online store. But, you know, in reality, you have to grow a protein into a solution. You have to make it very saturated and you have to make it crystallized. I think it could be like similar way that you crystallize like sugars or salts, but I'm not the expert in, you know, the X-ray crystallography. I have a friend who is also an expert in this field, and he said it's super, super difficult to get the structure because, you know, you have to get the structure in a very good shape, and it could take months, and it's also very expensive to do it. And once you get the structure, you can shoot an X-ray beam, and then you turn the structure around, you get this kind of diffraction pattern. And that pattern, you know, you have to decode that with a sophisticated computer algorithm in order to get the final protein crystal structure. This is one of the most, you know, difficult way to get the crystal structure, but it's also one of the most accurate way to visualize the protein as well. So this is very advanced technique, but it's been used for a few decades so far.
Ice, may I ask, within your presentation you had some lovely light and shade and some emotion. How important do you think it is that science provides hope at a time like this?
Well, I think science is very important to providing hope, because if we don't know the science, we won't understand how to save our life, how to deal with this situation. And, you know, if we don't know the science, we won't really know that the coronavirus could not be you won't know that you can use the alcohol, right, to save ourselves from infections, you don't know that you have to wear a mask. You won't know that you have to get the vaccine. You won't know even how the vaccine can be created. So the basic understanding and good understanding in science is going to help us survive. And everyone can be the heroes in saving this situation by following the guideline of the WHO or the scientists or the doctors.
Thank you so much, Ice. Lots of love for Ice and Thailand. Lots and lots of flags of support. Lubov says, loves the props. Katanyik is grateful for your devotion and your determination, Ice. Joanne says, great use of X-ray crystallography as part of it. Scott Bleakley says, ice clear presentation. See what you did there, Scott. First pun points of the evening for you. Mink says, good job. Great stuff. Keep the comments coming. Thank you so much. Next up then, let's get straight in. Next up is Eman from Egypt. Eman is a dentist, and her medical background has made her passionate about exploring behaviour. She's also passionate about writing and public speaking. So FameLab brings those two together, those two loves of hers. Please note, Eman's presentation contains discussions which some viewers may find upsetting. Eman's 3 minutes starts when she does.
There's this young lady, we call her Amy. She used to be smiley and breezy until she got sexually assaulted on the bus to school. She tried to repress that trauma, but eventually had to face it. And when that happened, all she feels about herself, her body, sex, safety, all changed dramatically. Now she is too terrified to even get on a bus to school. But how can a bus get to be so scary? Inside our brain, information is passed along as electric signals from one neuron to the other via neurotransmitters, and the strength of that signal depends on how much neurotransmitters and how many receptors for them. So when we are in danger, the amygdala catches really quickly on that danger signal and boom, fear response. And we all know how that feels. Sweating, heartache, stomachache, it doesn't feel good. So when the event is high impact, the amygdala is now starting connecting cues that are actually safe with danger. That's called fear conditioning. With Amy, that cue would be the bus. It's now a trigger for the fear response of hers. Every time she tries to get on that bus, she will feel all this trembling pain on the inside and eventually gets off. But good news, safety can be learned as well. If the ventromedial prefrontal cortex, that's part of our thinking brain involved in emotional regulation, together with the hippocampus, that's the memory house, if they both send signals to the amygdala saying, all right, we've done some thinking and turns out buses are actually safe. That way, a new memory can be formed, and that new learning is called fear extinction. However, fear memory is much more hardwired and powerful than the fear extinction. And this is why so many people with trauma might have the most difficult time trying to remember what safety feels like. So yes, Amy is now able to get on the bus again, but she is still lost inside her body. And that's not just Amy. Recovering trauma is a long journey, and you might need help on it. And by the end of it, hopefully, there will be no guilt, no shame. There you may find your body once again and fully reclaim yourself.
Eman, thank you. You've talked about it taking time for this process to happen. I wonder how much time are we talking, and what does that depend on really in each individual person?
Well, to answer that question, we need to understand that trauma is really a subjective experience, and this is probably one of the reasons it is a difficult phenomenon to study. So it might take a lot of time for people, for some people, it might take shorter for others. It really depends on how the person receives that and how their history, because it's been seen that when the trauma has been experienced in a very young age, probably the repercussions of that is going to be much more devastating and much more difficult to recover, but you know, there are still, of course, there is still a possibility to process that and get over, of course. And this needs the right, you know, handling of the situation, and it also might need the help of a professional. And so, yeah, am I missing anything about your question?
That answers it beautifully, thank you.
Can I build on that, Eman, and just ask you, so what is the therapy that helps this fear extinction to happen?
Basically, it's exposure therapy. Being exposed to the same cue that has been conditioned to fear, but exposed in a safe environment, most probably helps this extinction process to be further deepened and empowered. And actually, I want to take the time to mention here that the story I am using is not actually of my own imagination. It's a story from a series called Sex Education. I just didn't want to alienate people who didn't watch it. And I chose it because at some point, Amy, the victim, she actually managed to get on the bus because her classmates and the school friends, the girls' school friends, they all gathered with her and they came to the bus that they don't normally take, just to support her and give her the safety that she needs to get over it. But also, I intended to use this story because, yes, she got over one part of the trauma, but she still had a lot more to still process. So.
Thank you so much for this lovely talk. And I was wondering, what can we do in our daily lives to protect us for this case, for the trauma?
To protect against trauma, it's difficult to say because, as I told you, trauma is not the event itself. Trauma is how it affects us, and it has a lot to do with it. Our age, our background, how safe do we feel in the close proximal circles. There's a lot to it, and I don't think the point is to protect against trauma. The point is to know how to move on from it after you absorb it and accept it and be compassionate to yourself in the process.
Thank you.
Lots of support for Eman coming through on the live chat. Lots of flags, of course, for Egypt. Galia says, so good to speak about fear and how to overcome it. Mona says, so proud of you, Eman. Lubov in Bulgaria says, powerful talk. Thanks for spreading the news. Aya says, fantastic. And lots of people saying, you're a star, Eman. So yeah, thank you so much for that. I realise I haven't told you about the prizes. Both runners-up and the audience winner receive a prize for up to £200 to spend on something, whatever they want for the next steps of their science communication journey. It could be kit, it could be training, whatever they fancy. And the winner receives double that, £400, or of course, the equivalent in their own country. Plus, they all get the trophies. I've put all four of them around me now. Onwards then to our fifth international finalist. Our fifth international finalist is Jerzy from Poland. Jerzy is an architect and a researcher at the Faculty of Architecture at Wrocław University of Science and Technology. He is a big fan of paper and has plans to build a fully usable paper house. Jerzy's 3 minutes starts when he does.
When I was a little kid, I loved building houses out of cardboard boxes. Today, when I'm a little bit older, I still do it, but on a bigger scale. I have created a house for our victims of natural and human-made disasters. And the number of those people reached last year 150 million. The structure is made as a flat package that can be transported to the site and then folded into the form of a house. But actually, why should we use cardboard or paper, because they are exactly the same material, to build houses? Building industry is one of the most energy consuming and waste generating sectors. It is responsible for 24% of the non-renewable material extraction and 36% of global energy consumption. So if we take paper, we can look at it and it looks like a portion of spaghetti that was poured on the plate and left to dry. The structure of paper looked like the noodles of spaghetti, and the cellulose fibers which are in between create hydrogen bonds. Cellulose is one of the most natural polymer in the world, and its resources are inexhaustible. Paper can be recycled up to 7 times. So what can we do with one batch of paper? A house, a wardrobe, a box, a book, a newspaper, a tissue, and then at the end it will be all eaten by bacteria. If we compare 18 with 80 centimeter wall made of concrete, the first one is 2 times more thermally insulated. It is said that timber is a new concrete, but paper is an evolved timber. So if you will hear a story about the 3 little pigs, don't believe that the third one had a house made of bricks. It was paper. Thank you.
Hi, it was a really wonderful talk. Thank you. And I was wondering if why we use nowadays with this globalized world, we use the same computers and essentially dress the same things. But in housing, it's different because we still construct the houses the way we do it locally. Why do you think this is not the case with housing? I mean, why nowadays we are building houses locally, not so much though in the globalized fashion, say?
Okay, well, actually, like in the beginning of 20th century, there was this modernist movement, and it was recognized as an international style. So we do it in some way, in a global way. But of course, the best is to use the local materials, so you don't use energy from transportation. And but paper is actually the material that is super easy to produce if you have the infrastructure, and you can do it everywhere because it's almost everywhere accessible. So obviously we have our own characteristics, the climate, so you need to have like different type of roof and so on, which will make the architecture suitable for the local conditions. But I'm pretty sure that the style these days is more international and worldwide.
Thank you, Jerzy. That was a fascinating talk. Tell me more about this polymer that's in plastic. How exactly is it formed, and how does it make the houses waterproof? Okay,
well, in plastic or in paper, sorry?
You talked about the polymer in paper.
Yeah. Okay. The cellulose is the main component, and this is like the fibers that create each other the hydrogen bonds during the production once they are pressed and dried. And thanks to that, after we water it again, it can be recycled. And this is of course a problem for building industry. So paper is not recognized as a building material yet, because if the water would get there, that might cause some problems. However, there are several methods to impregnate it. You can do it in the mass when you produce paper. You can do it to coat it with some material. You can do it by for example, thermoplastic shrinking or thermo-shrinking sleeves, or you can do it like our friends from TU Darmstadt from Germany. They just build a core of the house out of cardboard, and then the outer layers are the layers that support the inner structure.
Jerzy, thanks very much. I wonder how much these houses would need to change, or the makeup of them would need to change. Say, for example, in Poland where you've got snow, in the UK where we've got plenty of rain, or perhaps in the Caribbean where there's more sun. How different would they have to be, and how could that be incorporated into the idea?
Yeah, then we would have to design maybe one system which can be flexible in order to fit into different climates. So, for example, there is a building in UK, in Westcliff-on-Sea, which is made out of several layers of honeycomb panels. and it's thermally insulated enough. The other example might be the German example, and the wall has 30 centimeters, and it fits exactly the local conditions. On the other hand, the building, for example, designed and built by Japanese architects Shigeru Ban for Kobe or from India, was made just out of paper tubes which are stuck to each other. But in the harsh conditions, they were filled with the crumpled paper. So depending on the climate zone, you can deal with the material and the building envelope.
Thank you very much, Jerzy. Some funny comments coming in. Lubov and Julia say that essentially the three little pigs need to think again. Science busts myths. Joanne says, such an interesting variety of topics in the final. I completely agree. They're so good. Katie says, great presentation. So much support. Thank you all so much for getting in touch on the live chat. We are now halfway through our international finalists. A reminder then that our judges will choose 2 runners-up and 1 champion, but you will also get to crown your audience favourite. The vote for that will open after all 10 finalists have performed. On to our next finalist then. Our next finalist is Bozhidar from Bulgaria. Now, Bozhidar, aka Dr. Bo, is an assistant professor in chemistry at TU Sofia. His research focuses on the very small nanomaterials, and he spends his free time building gadgets and tinkering with electronics and embroidering chemical clip art onto t-shirts and lab coats. Bo's 3 minutes starts when he does.
I have a service announcement to all of my bespectacled fellows. This is not a drill, folks. The winter is upon us, and with it, every time we walk into a room, we'll sink into the world of foggy gloom. Because our glasses fog up. And well, that's annoying, but why does it happen and what can we do about it? Well, I am a surface chemist, and even though I'm not a shallow person, for me the explanation and the solution to this problem is all about what's on the surface. The surface of my glasses, that is. When we come from the outside, the surface is cold, so the dew point of water nearby is lower. The attraction between the water molecule will overcome their thermal energy, so they will start to bunch up and attach to the surface. We call this condensation, you know it. So initially water will form very nice molecular layers, but at some point it will be in a very tough political situation. Because on the surface it's bordering 2 very foreign and unfamiliar worlds. That's the surface itself and the surrounding air. And when you make a border, of course, this takes energy, and sometimes even tensions may arise. We call this surface tension. So water will do the political choice to decide to expand its border to the direction of the lower surface tension. If it picks the air, then droplets will form and the surface will fog up. In order to avoid this crisis, we need a chemical ambassador to warm up the relation of water with the surface. And in surface chemistry, we use these guys. These are the hydroxyl groups. These are like little molecular fragments of water, which help it recognize the surface as a friendlier border. So in this case, it will spread across the surface and a transparent uniform layer will form. So, bye-bye foggy glasses. Well, that's amazing and you can see how well it works, but you might be wondering, what's in this for you? Probably you don't wear glasses at all. Well, the ability to control how water behaves on a surface is important in all sorts of applications, and nature knows best. For example, the lotus flower has evolved a very water-repelling surface at its leaves. This way it will form large droplets which can roll down and collect the dust. In the Namibian desert, there is a beetle that has engineered a complex patchwork of water-repelling and water-attracting surfaces at its wings. This way, it can gather the moisture from the air and survive in the drought. In my field of surface chemistry, these types of amphiphilic coatings are just emerging and they are the hottest research topic, but we still have to learn from nature. Finally, science, it helps us not only clearly see our immediate surroundings, but also open a window to the future, where the possibilities are many if one has imagination. Thank you, guys.
Thank you so much, Bo. As a glasses wearer, I did very much enjoy your presentation. My question is, you said that your science has applications way beyond glasses. So what do you think is going to be the impact of the work that you're doing out in the real world?
Well, there are lots and lots of applications of this, and the most important ones are being able to create self-cleaning surfaces, which will take less energy to clean. And last time I talked about the self-cleaning windows, but there are some other great examples. For example, I'm a huge fan of the work of Professor Ivan Parking from the UCL in London. He makes like self-cleaning fabrics which can repel paint, etc. And also in the UK at the University of Durham, there was a project to make a water gathering layer which can be used in some regions of the world where they don't have water, just to extract the moisture from the air. So there are many, many applications, and some of them are extremely important. The way I pick this topic is, it sounds very boring, but actually it has an impact.
Could you use it on solar panels?
Yeah, of course. Cleaning solar powers is something that takes a lot of energy, and it's very important for them so they can have the maximum energy generation. So yeah, they already have a product like this.
It sounds anything but boring, Bo. Thank you very much for that. You talked about nature being important, and I wonder how much collaboration there is between you as a chemist and the biologists.
Well, there is an entire field called biomimetic surfaces where chemists, surface scientists, material scientists and biologists, they work together. They will study, for example, the wing of this Namibian desert beetle, and they will try to replicate the pattern. Or, for example, make a very good adhesive piece of tape inspired by the gecko lizard just by the structure of its feet, you know. So, yeah, there is collaboration. Science is becoming so... It destroys the boundaries that I was talking about here. So, yeah, thank you.
Thank you so much, both, for this nice talk. I was wondering if it's possible to use this in this interior painting that we use in our houses in order that prevent mold to appear or something.
Yeah, but that's related to my other kind of research, the work that I do on photocatalysis, where if we add some material, which is a photocatalyst, to the paint, it can use this light from the lightning inside and just destroy molds and viruses and bacteria. And that's already done, so it can be applied there as well. Yes.
Wearing masks and glasses and this condensation problem, have you got an immediate tip for us on how to prevent condensation whilst wearing a face mask?
Yes, just try to push the nose bridge very hard on the upper part of the mask, and this way the vapors, they will go around. So that's my suggestion.
Little bonus tip there at the end. Thank you very much, Bo. Bonus tip, I've just realised that one. Alexandra says, look at the blackboard. Some subtle hints there about who to vote for. Relitza says, Bo is amazing both on the surface and in depth. Moon says, we love you, Bo. Go, go, go. Darby says, excellent visual aid. Lots of people loving the giant set of glasses that you had there. I thought that was absolutely brilliant. Lots of people loving the enthusiasm. Dorina loving the puns. Claire says, fantastic talk, Bo. Love the idea of chemical ambassadors. So lots of love there. Thank you so much for the comments. A reminder, the audience vote will open after you have seen all 10 finalist performances. And while you are casting your votes, I've got a few things to keep you entertained. The first is a celebration of the 15-year partnership between Cheltenham Festivals and the British Council, and a look at what FameLab International has achieved with a special guest musical performance. And the second is a live chat with an honoured guest who many of you may recognise. It's last year's winner is going to be joining me very soon. All of it, we like to say it's a Super Bowl halftime show, but at full time and for science. Onwards though, next up we have Alex representing the Netherlands. Alex is currently doing her doctoral research at Amsterdam UMC, and she says that by day she studies autophagy, that's our body's way of cleaning out damaged cells, and by night she studies The Princess Bride. She is an avid fan of cycling and sustainable living as well, in fact. Alex's 3 minutes starts when I hit this button.
Imagine you're sitting at your desk and somebody knocks on your door and they hand you a bucket that's filled with fresh human skin? Ugh. Welcome to our lab. where we receive donations of human tissues like skin that's left over after surgeries, and it would otherwise be thrown away. This may seem disgusting or gross, but it's actually amazing because human tissues, those are powerful tools for studying human viruses. Myself, I study dengue virus, HIV, and SARS-CoV-2, and I'm primarily interested in understanding how our immune systems see these viruses, so I can help defeat them. And in order to understand this, I'd like you to imagine a chess set, Queen's Gambit style, but I'll help. So when a virus plays your body in biological chess, it's playing white, making the first move by infecting you. But let's focus on the black army, your immune system. We often think of our immune systems in the singular, but they're actually composed of a lot of different moving pieces. For instance, we have the pawns. Those are the dendritic cells, which patrol the perimeters of your body, like in the top layers of your skin. And they may seem benign at first, but as soon as they notice an invader entering the field of play, they spring into action and alert their comrades that it's time to prepare for battle. Next, you'll see cells that, like bishops, roll out predictably, but quickly, to hopefully give our side, the black team, an early advantage. Those ones are called, and I kid you not, natural killer cells. Next come cells that, like the queen, can make efficient and far-reaching attacks. Those are the B cells, which produce antibodies that spread out over the entire field of play, and they pick off viruses, one by one. Now, this isn't a comprehensive overview of all of the immune cells or all the chess pieces, but you get the idea. Each of our immune cells is unique, and they all have a role to play in the battle of the host and the hijacker. And that brings us back to the skin. Because often in the lab, scientists will only look at how immune cells or viruses interact with immune cells one cell type at a time. And that gives us important information, but it doesn't give us the same insight as if we look at the whole board. And that's how what we do is different. By looking at the whole chess set, we can see how viruses react when they're faced with an immune army. And by using human tissues to do this, instead of lab rats, for example, we're using the most realistic possible system to assess this battle and see where we can help these guys out. Because our ultimate goal is to find new and more accessible ways to treat the viruses we study by giving our immune systems a little help. So those buckets of skin, they're not just a great conversation opener. They're also our way to say to the virus, check, mate.
Thanks so much, Alex. I wonder whether I can ask, how much do we know about how the natural killer cells talk to the B cells, the bishops to the pawns and so on, and the impact of that?
Yeah, really interesting question. So to be honest, I actually don't focus on natural killer cells in my immune system. I chose them because of in my immune system. I hope that's using natural killer cells. In my research, I chose them because of the awesome name. But what I wanted to showcase is that there are different sort of sectors to the immune system. So natural killer cells, those are the first defenses, part of the innate immune system that kind of always responds. And all these different cells in the innate immune system, they basically send out biological letters called cytokines that will spread out throughout the body. And then they activate specific second responders like B cells and also T cells in a way that's suited to whatever the particular opponent that they have detected is. So actually it really depends on whether it's a virus, a bacteria, a creepy crawly parasite, and it's a huge research field. So we actually do know quite a lot. Yeah.
Alex, thank you so much. That was fascinating. So one thing I loved about what you had to say was how you looked at the whole immune system. So you weren't looking cell by cell or at the different elements of our immune system. How does that require bringing together different disciplines within the work that you do, or is it just a different frame that you set the research in?
Yeah, I would say it's a bit of both. So I like to say that my research is at the intersection between infection and immunity. Oftentimes people will focus more on viruses or more on the immune system, and we really try to bridge the two and look at the connections. And in terms of setting the frame, yeah, I would say I mean, the chessboard does have some limitations. I would say we're using as much of the chessboard as possible in an ethical and, I like to think, quite environmentally friendly way as well, kind of turning this trash tissue into treasure. So we're basically setting the frame as wide as possible and bridging all sorts of different fields to get the most realistic answers, to get the closest to the truth.
Thank you.
Thank you so much, Alex, for this wonderful talk. I have something in my mind that I am curious for a long time. For example, when we are infected with COVID, how many particles do it's necessary to make us ill? I mean, if there is just one particle, most of us will be able to, I don't know, to kill it. Do you have an idea that this is known or not?
This is a super interesting question that people are still looking into for COVID. What's interesting about this is it's completely different depending on the virus or the bacterium. For instance, I'm quite sure with Mycobacterium tuberculosis, causes tuberculosis. If you get one bacterium, you can develop tuberculosis. With most respiratory viruses, like SARS-CoV-2, you usually need a bigger dose. Like if you walk through a cloud of particles from somebody's cough, you're more likely to get infected than if you're outside and there's one lonely virus drifting along that gets onto you. Usually your immune system will be able to clear that for viruses related to SARS-CoV-2 at least. But that's still an ongoing question for our latest SARS.
Thank you so much, Alex. Thank you, judges, for the questions. Lots of live chat action going on. Jessica says, go Alex. Tara says, great prop. Lots of people loving the chess set. Kanoto says, props have come out to play today. Edgar says, what an efficient way to explain our immunology. Brilliant. Mahmood, who was one of last year's finalists, is watching, says, love the chess analogy. And Scott says, checkmate. So we only have a trio of FameLab finalists left. And the first person in that trio is Letago from South Africa. Letago is a master's student at Northwest University investigating the impact of climate change on farmers in a province of South Africa. When she's not doing her research, she loves reading, a whole load of different outdoor activities and working with youngsters in her local area. Letago's 3 minutes starts when she does.
My favorite place in the world is at 23 degrees, 49 minutes, 58 seconds south, 30 degrees, 9 minutes, 48 seconds east. You're probably wondering what these are the coordinates for. This is a town called Tzaneen in the Mopani district located north of the Limpopo province here in South Africa. Why is this my favorite place, you might ask? I love a good fruit salad, and this place is known for its fruit industry as it is one of the major producers of avocado, papaya, and mangoes in the entire country. But my favorite place is in a bit of a crisis. In the last 20 years, it has experienced over 45 extreme weather hazards associated with rapidly changing climate in the form of hailstorms, floods, droughts, veld fires, and even secondary impacts from tropical cyclones off of the neighboring Mozambique. The impact of these hazards has been seen in agriculture through decreased crop production, animal disease, poor crop health, and land degradation. Now, climate change is a naturally occurring phenomenon that often balances out in nature. However, human activity has increased its impacts in both frequency and magnitude, and unfortunately, man cannot be saved by Mother Nature indefinitely, which is where my research comes in. Smallholder farmers in the Tzaneen area use methods that are inexpensive and efficient that have been passed down from generation to generation in order to combat and adapt to the impacts of extreme climatic conditions. Now, were you aware that you are able to store leftover seeds from your harvest in a little bit of firewood ash, and this allows you to keep the seeds for more than one season? The beauty about firewood ash is that it also acts as a pesticide. So over and above preserving the seeds for you to plow with again next season, it also keeps all the pests away in case you want to consume it at a later stage. As farmers residing in a hilly area, are you aware that you are able to plant your crops along the steeper gradient of the slope? This allows you to retain some of the water from flooding, and it reduces the risk of soil erosion. But the real beauty with this method is for farmers on flat-lying areas, because you can create your own contours. By piling up rocks found all over your farm and filling in the gaps with soils, you are able to create bumps or barriers that act as contours that will also help in terms of soil retention in the case of soil erosion. In the Zanin area, indigenous knowledge has proven to be helpful in the achievement of food security at a household level. So can you imagine the impact it would have if we're able to integrate this wonderful knowledge system into development of sustainable adaptation policies to help assist rural communities which are vulnerable to climate change hazards? Thank you.
Thank you so much for this wonderful talk. I was wondering, what is your opinion about organic farming? I mean, somehow this organic farming, somehow they do not accept so much these scientific breakthroughs science bring for farming, right? And what's your opinion on that?
Absolutely. I think organic farming alone at a larger scale is not as sustainable. I think the idea behind indigenous knowledge as something to integrate into scientific knowledge, I believe that the two will marry and work well together. When you're working at household level, it's easier to apply organic farming and more indigenous-based methods. But when you're working at a larger scale, for example, if you're going to use biopesticides, how many insects and leftover leaves do you need to grind in order to fumigate an entire farm? So with respect to organic farming at a larger scale, I think that there is a question of sustainability. However, at household level, it is very much effective.
Thank you so much. I really enjoyed your talk, Letago. My question is around how we get those scientists working on the food system side and on the climate adaptation side to really take this indigenous and local knowledge seriously. The examples you gave are really impressive, but how do we get them to respect these local solutions as part of what they also need to be advocating for?
I think it is already something that is growing. Looking at COP26, indigenous knowledge is already something that is being thrown out there. There is a lot more recognition that is going into it. And I think as up-and-coming scientists that see the value of it, we are the ones that have that burden to say that this is something that can be looked into, this is something that has worked, because indigenous knowledge has so much value. It wouldn't be knowledge if it wasn't effective. It wouldn't be indigenous knowledge, it wouldn't be passed down for generations to generations if it did not have value. And so that is the starting point for that, to say that as up-and-coming scientists, we see the value in this. And like I've said, I've tried it out at a smaller scale, at household level with smallholder farmers. So then this is our chance to then expand it and write papers, put out research, and advocate for its inclusion.
Letago, thanks very much. It's fascinating. I wanted to talk about those small householders, and when you go to talk to them, how keen are they to share that information, and how aware are they that this could be game-changing?
I think they're very much aware of exactly what is going on. The one thing that people always ask me is, are farmers even aware that there is climate change? And I say a lot of farmers perceive climate change on two bases, temperature and rainfall. So they will tell you that rain does not fall in the season that it used to, or that temperatures are increasing at a rapid rate. And so they are very much as keen to find solutions to this as we are. So that makes it easier when approaching them to say, what changes have you noticed? And what are you doing about it? And how can I marry your knowledge with mine to better find sustainable methods? And it's been amazing because they're really open to saying, okay, this is what we know, because we're also including them. I think the beauty about science communication is the inclusion of the people, because once they understand what your research is, then they're not as intimidated by the science anymore. And that makes the collaboration that much easier.
Thank you so much, Letago. Lots of comments on the live chat. Also, lots of people praising the judges' questions, which is always lovely. Great job by the judges. I must say that, because that is a very tricky job. Edgar says, very impressed by the adaptations happening in farming. Surfiro says, good topic, easy to understand. Ketchepoe says, well done, Letago, very interesting talk. Irene says, go Letago, from farming to fame. Lots of people faith supporting from South Africa. And Tara is watching. Hi, Tara. One of our judges indeed, saying absolutely thrilled to be a FameLab judge. Such amazing science communicators among this year's finalists. Absolutely. Our penultimate finalist then is Danny, representing Mexico. Danny describes himself as a fellow earthling who likes nothing more than walking outside his home and watching the fruits growing on the trees and hearing the bees buzzing around and drinking water fresh from the stream. His 3 minutes start when he does.
Amy Stewart, the author of Earth Moves, said they don't sing or chirp, they don't gallop or soar, they don't make tools, hunt, or write books, but they do something just as powerful. They consume, they transform, they change the earth. She was talking about the earthworm, and I'm here to tell you that she was right, and that we've been literally stepping over a earth-changing solution that, if given the right attention, could be a game changer for the United Nations race for sustainability for 2030. Like Stuart before us, our team and I today realized that the ability of the earthworm to transform waste into decay, what we think is garbage and useless, into something amazing, will reconstruct global recycling, be able to revitalize soil with microorganisms, and help cultivate a sustainable future that'll get us to 3030. We want real global solutions, right? Well, it's time to start collaborating with the best of the best. And who better to help us to care for the earth than the earthworm, who's been doing it for hundreds of millions of years? You see, we are so stressed and concerned about what's happening with our planet. We're running around having global conferences, billionaires willing to donate billions, major reconstruction projects on the table, but we've been doing this for years. It should be clear now that real global solutions are not found in the flash and buzz of the world stage. Real answers will not be heard in the roaring and screaming of crowds. But one real solution can be found in the amazing intestines of the earthworm. Wait, what? Yep, you heard it here. Producing earthworm poop is a real global solution, and my team and I have developed a smart, semi-automatic vermin composting system that makes playing with earthworms fun, but more importantly, impactful. And guess what? It can be done in the comfort of your home. Not all solutions are found in the billions of dollars, but in the billions of earthworms. And it gets even better because you and I really don't need to do that much, thanks to the intestines of the earthworm. On a daily basis, you and I just need to feed them this. Remember when I told you that earthworms eat waste and decay? Well, by simply feeding them some of our inorganics and organics, they will produce the most important poop of our lifetime. And if mass produced, it would create a transformation that we'd all see in our lifetime. Just to give you some facts so you can see how incredible this is, vermicast is a renewable resource, super important. Once in the soil, it brings rich biodiversity back into the soil, giving life to this earth. Are you excited yet? And it's able to help bring sustainable infrastructures where everyone around the world benefits. Ladies and gentlemen of the world, I present to you lombriz, the earthworm, a real climate solution. And I invite all of you to begin working with them so that all of us can help our local farmers to be able to bring life back into our earth, and most importantly, share in a solution that we all need. And it comes locally in a small package in the dark, quiet soil where we find the unsung heroes doing what they do best. And that is consuming, transforming, and together changing the earth. Thank you.
Danny, thank you. I always thought I had great respect for earthworms, but now I'm just totally a fan. But tell me a little bit more about the impact that they can have. So I get that these earthworms have the ability to transform waste into compost, but using that compost at a global scale. Tell me more about that and how it creates impact.
Well, I mean, the big thing is chemical fertilization. And so if we can start phasing out chemicals, that instantly, automatically, we're going to decrease the chemicals that go into our food, which is a big cause of cancer, for example. A lot of the chemicals that are actually put into the plants are not absorbed. So there's a lot of runoff. So that goes into our water systems, it goes into our air. So by just simply giving earthworm casting or vermicast into the fields, we eliminate air, water, and soil contamination.
Thank you.
Danny, may I ask, can you tell us what's on that tray that you held up?
Oh, of course. My breakfast. So we have melon, we have cucumber, we have paper, and then toilet paper roll. It's amazing how many things around our house can be that earthworms can eat. So that's why I say organics and inorganics. There's things around our house that we do not need to throw in our trash. And one of the most important things is that organics, it's not trash. It's something that the earthworm can take and transform and make it into something amazing.
Thank you for this nice talk. I was wondering if it's possible to recover soil that it's already damaged by chemicals. I mean, and for how long do we need to wait for these worms to recover this kind of soils?
Best question. Absolutely. We can absolutely regenerate our soils. That's the beautiful thing about vermicast. If you go into, nearby Puebla, a lot of our soils are just filled with metals. But as soon as we start putting vermicast, it begins to restore all of that. And so within a couple of years, you can have soils that have been dead now producing crops again. Because again, what vermicast does, it brings back life into the soil. It isn't just about restoration, it's about giving life into soil, which at the end of the day is what we need to be able to produce quality crops.
Danny, may I come in with another question? You talked about this being scaled up. I wonder, what would that look like in the landscape? What are you envisaging?
Perfect. Yes, so my team and I have developed a system where you don't have to be an earthworm expert. The system does everything for you. It's automatic. It controls temperatures, pH levels, humidity, all of that. And so you put this in your home or your business or in a cafeteria in a big factory, and all you have to do is throw all of this into this smart vermicomposter. So just imagine millions and millions of homes and businesses around the world not throwing this into the garbage, but throwing it into this smart vermicomposter. And there's where we have the mass production of vermicast worldwide.
Can I ask you one last thing? Vermicast. What is the first word? I don't understand it.
Vermicast.
Vermin or verming?
Vermin. I'm sorry, that's my accent. I'm sorry. I'm so sorry.
No, I got it. Thank you.
Worm poop. Worm poop. That's even
got
it.
Vermicast, aka worm poop. Lots of love for the worms on the live chat. Good work with the emojis on the worms as well, you lot. Lots of people saying, Sungay from South Africa says, earthworms? FameLabbers are awesome. You bet they are. Galia says, great presentation. Enthusiastic, clear and engaging. Well done, Danny. Katjanik says, worms are the world's saviour. Thank you, Danny. Marco says, making such a small change can make such a great impact. So yeah, lots of food for thought, that one. Just one talk left, sadly, but then the audience vote will open. Our final finalist is Hugo from Switzerland. Hugo is currently doing a PhD in statistical physics and machine learning at EPFL in Lausanne, Switzerland. And when he's not at the office, you can usually find him wandering around the city or on the top of a mountain. Hugo's 3 minutes starts when he does.
And artificial intelligence, or AI, is an algorithm that can learn to do amazing things, recognize people, animals, and photos, find new carbon-capturing materials to combat climate change, or even help detect cancer. How do they learn all this? Data. Big data. An AI learns by analyzing data sets, so for example photos, and the more data it analyzes, the better it learns. But there's a problem. Because the more data an AI has to analyze, the more time it will take, and the more computer power it needs. And for some applications, this means a huge supercomputer working for months, consuming gigawatts of electricity. That's just not sustainable. Can we do something about it? Yes, we can. And to give you an idea what, let us play a role-playing game. I'm with a data scientist and you play the role of the AI. And your mission today, should you choose to accept it, is to learn to distinguish photos of kangaroos from photos of wallabies. That's a typical task in data science. I have here a data set of 4 photos. I show you each photo, tell you whether it's a kangaroo or a wallaby, so that you will progressively learn to differentiate the 2 animals. All clear? Okay, let's go. So this is a kangaroo. This is a wallaby. Hold on a minute. In that photo, actually, have you in your learning process? No, right? The small silhouette in the background far away, you can't see much. This data is essentially useless for you. Next is another wallaby. This, on the other hand, is very useful data. You can see exactly how a wallaby is built. And it is shorter, rounder, and cuter than a kangaroo. Very useful. Last photo, another kangaroo. Again, blurry photo, useless. So, out of the 4 photos I had in my dataset, how many were actually useful for your learning process? Only 2. And I'm not showing you the other 2 photos, well, you'd have learned those as well. This is true for any dataset in data science. Only a fraction of the photos of the data is actually useful. In practice, researchers in computer science like myself are developing algorithms to identify which data in the dataset are useful, which are useless. Once useless data have been identified, they can be thrown away. This way, the AI will have less data to analyze. Therefore, it will use less time and less computer power. What's more, since the data I throw away was useless anyway, well, the quality of the learning will remain very good. Because it's really as people say, learning is not about quantity, it's all about quality. Thank you very much.
Thanks so much, Hugo. I guess I'm going to ask about applications. Where do you see this being most useful?
Oh, for example, some people are doing research to find which materials could be great for building a quantum computer, for example. And the fact is that you want to show your AI for it to learn to predict if your material is good or not. You want to show it the material and say, this is good, just as I did for the kangaroos and the wallabies, or this is not good. But you have to know before if a material has good properties or not. And for that, you need to run lengthy computer simulations. So you cannot do it for any data set of material. You can't do it for tens of thousands of material. You have to pick a certain number of material which you think are interesting, only make your computer simulation on those, and then give it to your AI to learn. And that's why you have to reduce the size of the data set, otherwise you have years and decades and centuries of computer simulation ahead before it can actually give your data to your AI.
Thanks, Hugo. So this is fascinating how you use AI to pick out the useless data. Does that process itself not also use a lot of energy?
No, because we're designing these algorithms specifically to run very fast, otherwise I agree with you, it would completely defeat the point. And also you may think that it's not about also just learning from the data, it's also about storing the data. And you know that these data centers consume lots of energy. It's 2% of the energy consumption, the electricity consumption in the US. And worldwide, I don't know if you can guess the figure, how much electricity we're consuming for data centers in a year. But it's 200 terawatts, 2 with 14 zeros afterwards, 3 times what Switzerland is consuming per year. So just having smaller data set can help also cut this energy cost at the level of the data centers.
Thank you, Hugo. I was wondering how will be data science when we have the quantum computer? If we will have someday, I mean, maybe we don't have all these needs of compressing data and so on, because it will do everything, I guess.
Well, let's talk about it in 50 years. Of the time being, quantum computers, it's in embryonic states of research. It will be just very fast, and some researchers are already devising some machine learning algorithm with those quantum computers. But there will be a revolution, and as for any revolution, there's no telling what will happen. So I'm bracing myself for lots of surprising and lots of advances in science, and I hope you're as excited as I am.
Thank you.
Thank you, Hugo, our final finalist. Lots of people saying, Hussein was saying, Hugo's on fire. Amer's watching, our semi-finalist host. Hello, hello. Saying, fantastic, Hugo. Lots of people saying, well done. Galia saying, well done. That's it then. You have now seen all of our 10 FameLab International finalists perform. As I've mentioned, those talks and the judges' questions after each one were pre-recorded last week. And after all finalists performed, we sent the judges off into their own digital room, virtual champagne in hand. We treated them. And they found it. They then had to battle it out to work out, okay, who's going to be our 2 runners-up and who are we going to crown the FameLab International Champion 2021? As I also mentioned, they did not tell the finalists and they also did not tell me. So we will hear that together very, very soon. However, there is another title up for grabs, and that is the FameLab International 2021 Audience Favourite. That is out of the hands of the judges and it's into the hands of you. And the vote for that is now open! So to vote, you need to go to this link here. You need to choose your favourite 3 finalists and rank them. So you rank your favourite as number 1, your second favourite as number 2, your third favourite as number 3. We are going to give you 10 minutes to do that. So I'm going to start the clock on that now. I've got lots on the way to entertain you while that happens. We're going to start with a quick recap of all the finalists. Here we go. So, who are your top 3 and who is your favourite? This is the time to cast your vote. And while you're doing it, I have 2 more things for you. Firstly, this is the final year that FameLab is going to be produced through a wonderful partnership between the British Council and Cheltenham Festivals. To celebrate that partnership and everything it has achieved, we've got a message from Adrian at the British Council and Ali at Cheltenham Festivals. And then, secondly, we have a very special musical message from FameLab alumni and semi-final host Amer Maguire. You're in for a treat. Here we go.
Hi, I'm Adrian from the British Council, and I've had the privilege of overseeing FameLab International for the last 5 years, and its impact has been massive. Each year, Egypt attracts over 1,800 entries. We've had new science communication organisations established. We've influenced government policy and practice. Festivals in Bulgaria, Athens, Northern Ireland, all influenced by FameLabbers. We've also had TV coverage in Brazil, in Portugal, and numerous countries. And many journalistic opportunities for entrants working for newspapers or doing their own books. But for many, the impact is on the individual, and that's just as important. the chance to go on a personal journey, challenge yourself to present about science, and return to your research or workplace with a fresh perspective. Now, Cheltenham Festivals gave us this opportunity, the catalyst for an international programme, so we must thank them for the 15 years of support, input and dedication to making FameLab what it is today. Now, outside of that, there are the in-country partners, the in-country teams, the participants, the trainers, and all organisations that support the FameLab family. And we mustn't forget them, because you're all part of this too. So this might be the end of an era, but it's certainly not the end of the British Council's mission in seeing science as part of culture. We wish Cheltenham Festivals well with the next steps with the programme, but we want to stay in touch. You're still part of the FameLab family, and together we can see what we can achieve. Thank you very much.
Hi, I'm Ali. As Adrian said, FameLab has a place in the hearts of thousands of people across the world, and we want to thank the British Council for such a fruitful partnership over 15 years. We're looking forward here at Cheltenham Festivals to continuing the legacy of FameLab, creating an active international community of science-related researchers who are confident and skilled at communicating their research with the public. We'll continue to support this next generation of science communicators by establishing an international network of trainers and institutions sharing resources, best practice and opportunities to support science communication. And to celebrate the impact and legacy of FameLab over the last 15 years across the world, we're delighted that FameLab alumna and semi-final host Eman has summed it all up with her guitar.
It's been 15 years of talks and cheers and making friends with people we would not have met, yet won't forget. A global club just filled with love. It's FameLab International FameLab. It's fantastical FameLab. Isn't just a competition, it is friendship, science, art, it's tradition, and as long as we're still here, FameLab will live on. A science communication competition, like a sort of scientific, even better, Eurovision and Olympics. For science stories with clarity, charisma, content filled with allegories. People from all across the world stand upon a stage as they hold their nerve, tell us of the science that sets their soul alight. I've never met so many stars who burn so bright. It's FameLab International FameLab, and it's fantastical FameLab. Isn't just a competition, it is confidence, learning, heart, it's ambition, and as long as we're still here, FameLab will live on. The stories our scientists have told have been witty and emotional and wonderful and bold. No topic off limits, nothing you can't do. We've even had a finalist who dressed up as Pooh. And in 2020, an adorable prop. In the name of science, someone brought their dog. We've also had celebrities in the audience, I mean, like, how cool is that? We've even had the Spanish Queen. Egypt did their 10th finals at the pyramids, and our speakers have inspired science-loving kids all across the world, from countries far and wide. What I wouldn't have given to see FameLab as a child. Now, one of the best parts of FameLab is the master classes you experience to make sure you are faster at thinking on your feet, better at talking openly. The trainers are the masters of the Masterclass League. And FameLab all began in 2005, and then 2 years later internationals went live. Over 40 countries have been involved. It's crazy, amazing to see how it's evolved. It's FameLab International FameLab. It's fantastical FameLab. Isn't just a competition, it is passion, wisdom, fun, an expedition, and as long as we're still here, FameLab will live on. New colleagues and friends for life. Some FameLabbers find their FameLab husband or wife, and the family is going strong. Who's been involved? That list is long, from participants to global teams to partners were bursting at the seams. 200 organizations supported us, our adulations, and this will be the final year that the British Council are the partners here. But the FameLab spirit will still live on through Cheltenham festivals and this song. It's FameLab International, FameLab. It's fantastical, FameLab. It's just a competition, it's a worldwide family. My best decision, and as long as we're still here, FameLab will live on.
Oh my goodness. That was incredible. That was so good. So much love for that on the live chat. Benedict saying such a brilliant song. Moon saying this song is so sweet. I'm on a Zoom call with all of the finalists and everyone was absolutely loving it, asking where they can go and get the album. So that was absolutely amazing. Ema, thank you so much for making that for us. I think we all feel quite touched by that. Not long left, less than a minute left on the audience vote. So get stuck in if you want to get an audience vote in there. And while you do that, got something quite special right now. Please welcome, joining me live, it's last year's international champion, Surajit Majumba. So hang on, I've got to bring him in. Here comes the challenging bit. Surajit, hello.
Hello.
Hey, I've got you. How you doing?
Can you hear me?
I've got you. Are you well?
Hi, Greg. Yeah, I can hear you well. Hi, Greg. Very nice to see you back again.
It's so lovely to see you. Gosh, I remember last year like it was just the other day. So you won last year's FameLab International. What have you been up to since?
I'm continuing with my PhD, my research. That's the first thing for sure. I'm on with my research work over there, and along with them, I'm trying my best to find some time for my other passions, science communication being one of them. So yeah, I mean, we had some pretty nice events. I got to meet the finalists of this year's FameLab Swiss final, and we had a we got a chance to be at the British ambassador's residence in Switzerland. So that was a very special night. So we're grateful for that opportunity. It was nice to also be on the other side. So I was thankfully invited as one of the judges for the Lausanne semi-finals. And now I know how difficult it is also to be on the other side and select winners at every round. So that was quite some experience. So yeah, it's been on and off with this. So I hope to keep in touch with these activities. So yeah, overall very great. So I mean, I still don't believe it's 1 year. I mean, I just still need to remind.
How were you feeling a year ago? I've got all the finalists here, and some look nervous, some look chill. How are you feeling?
To actually revisit the feelings, right now I'm exactly the same room where I did my final talk of last year, to actually get back all those vibes. It's kind of a mix of different feelings, I think. Actually, I was excited, I was nervous, stressed, happy, and all of these stuff. So I can imagine probably many of them are also going through a plethora of different emotions, and it's good. You actually need to think how much you have to go through all these emotions at the same time. You can, but yeah, you have to.
Absolutely.
But, yeah.
I should say voting is now closed. It closed about 30 seconds or a minute ago. So
okay.
Audience
Voting
vote. It's currently being totted up as we speak. So last one then for me, Surajit, do you have any advice for anyone watching who would like to talk about their science?
Yeah, I mean, one thing which I realized from this experience of FameLab in general and science communication in particular is we should it's something we should take as a responsibility, along with doing science and doing excellent science. It's also important that we communicate it to the people, because at the end of the day, what we are doing is for the betterment of the society. So it's important that the society gets to know what we are doing. They're aware of the challenges we are facing, the improvements, what they need to do, where do they come into plan. And I think now, ever than before, I think is the best time for us to realize that, from what we saw from the pandemic, how important it is for accurate scientific information to reach people in the midst of so much of fake news. So that's it. So think with your head and speak with your heart.
Love it. It's been a pleasure to speak to you again. So lovely to have you on the final.
Same here.
Thank you so much, Surajit. Good luck in the rest of your science communication adventures.
Thank you so much. And good luck to all the finalists for the nice moments.
See ya. Thanks, man. Bye.
See ya.
Bye.
Bye-bye.
Right, the audience vote, as I said, is now closed. We are tossing up those results. I will be told who that is very soon. I cannot wait. We will reveal the FameLab International 2021 audience favourite very soon. The finalists are with me. Let's bring them up. You can have a good old wave. There they all are. There's our finalists with us live. Just before we hear the results and before we announce the runners-up, and then the audience vote winner, and then the FameLab International 2021 winner, let's hear from our judges about what they thought about all of our finalists.
Well, goodness me, what a wonderful way to spend a couple of hours. It's such an energising time. All of the contestants, all of the finalists have done such a wonderful job. You are all winners, that's the first thing to say. Really difficult decision. The variety is something that's really struck me, just in terms of the content, the use of props and so on. So creative, so well done, everybody.
Hi, I was really impressed by these nice talks. The level was really good. It was a really good, difficult job for us, the jury, to choose the winner. And yeah, the props were really incredible, and you did a really amazing job to present this online and directly through Zoom. So it was really incredible. It was a really pleasant morning today. Thank you.
And thank you from me also to all the finalists. You entertained me, you informed me, you did the hardest thing any scientist does, which takes all those facts and intricacies that you know about your science and paired it right back to just deliver your core message to us. And you did that with humour, passion, a story, and you made it relevant to our world and the challenges it faces today. So thank you. They were absolutely excellent.
Thank you so much, judges. You were also absolutely excellent. Here we go then. We are going to start with the judges' runners-up in no particular order. It's going to be over to Tara to announce the first runner-up. Got to make sure I press the correct buttons. Very important. Hang on. First runner-up.
This finalist hooked us in right from the start and in a surprising way, and then didn't quite take us where we thought we might be going, but displayed an absolute passion for their subject. They showed us how something quite unexpected can be one of the global solutions to one of the most pressing problems we face. Our first runner-up is Letago, representing South Africa.
Congratulations, Letago. I'm going to bring you up in the Zoom because I've just seen that amazing reaction. There you are. Let me switch to you. How are you doing right now?
Feeling amazing. Oh my goodness. Oh my goodness. Thank you so much. Oh my word.
Well, congratulations. It was such an interesting talk. I'm so interested in kind of what we can learn from the tools and the techniques that have been used for generations. Tell us a little bit about you and about your research as well.
My research is phenomenal. I come from Limpopo, which is one of the agricultural hubs of South Africa. So it's an amazing place for all the tropical fruit and vegetables you can think of. And so my research is about the people, you know. My slogan throughout the competition for people that have followed me has been science for the people, with the people. So I wanted to find out how the people are maintaining, you know, food security in such a tough time in Africa. And, you know, indigenous knowledge comes out top. So it's been such an amazing experience being a master's candidate and getting to go into the communities and speak to them about, you know, their everyday challenges, but also their everyday solutions. And I think science is much bigger than what we do in the labs, and going out into the community has been absolutely amazing.
And how has this FameLab journey been for you? How has it been being part of this new family of FameLabbers?
It's been ridiculously amazing. It's gone beyond my wildest imaginations. Honestly, when I first entered, it was because my supervisor sort of nudged me. So shout out to Dr. Sheldon Stradham. But everything after that has been 10 times more than I could have ever anticipated. The master classes have built me not only as a scientist and as a science communicator, but even as an individual. And so moving forward, I expect great things from myself and this entire panel of people as well.
Well, we cannot wait to see more of your science communication. You're a natural. And it's great that the judges voted you a runner-up. So congratulations. Nice one.
Thank you so much.
Bye. Oh, amazing. Okay, good. Cracking. Well, let's find out who the second runner-up is then. I'm going to go to Julia for that announcement. Here we go.
So this finalist was super clear throughout their presentation and hugely engaging. They drew us in right from the start, but halfway through or so there were some fantastic images that I have to say I will be trying to get out of my mind for some time. I was a little concerned at one stage that this person's co-star might be taking over, but they shied away from the advice never to work with children and animals. So I think you can guess now who I am going to be announcing as the next runner-up, and that is Samantha from Australia. Congratulations.
Yeah, nice one. Congratulations, Samantha. Let's bring up the Zoom. Hang on, let me press the right button. There you all are. Let's bring you up. Say hello to me.
G'day, how's it going, guys?
Very good. How are you? Isn't it 5 o'clock in the morning where you are?
4 a.m, yes.
Oh, even worse. Well, thank you so much for joining us, you absolute champ. So yeah, runner up. And as we saw, quite a lot of friends you had in the room with you, didn't you? How are they all doing? They asleep? Well, I guess not.
Yes, they got the night off, but I will be sure to pass on those kind words to all the amazing spiders and venomous animals that I work with. You know, I can't do my research without them, and I'm just so excited to get to share how cool these beautiful animals really are and try to help people appreciate them, not only for their importance in our ecosystem, but how they can actually help to save lives.
Yeah, that notion of using, spider venom, for example, to come up with new drugs. Is that quite a new idea, or is this something that's been worked on for quite a while?
It's actually venoms for drug discovery has been around for a while. The actually very first anti-high blood pressure drug came from snake venom. But spiders are a sort of a newer player in the field because the smaller size has made it very hard for them to be studied until the technology has improved in recent years. So we're really hoping to, show people that spiders are really holding the key to the world's most critically important medicines, and watch this space because that big huntsman or tarantula on your wall might be the one that can save you from parasitic infections one day.
I love the way you casually say the huntsman or tarantula on your wall. Fortunately, none here in the UK in my spare room, but maybe where you are. So final question then, what's this journey been like for you being part of FameLab?
Oh, it's been a wonderful journey. I think the best part for me has been just meeting the fellow participants who are all so talented and engaging and inspiring. And the research being done around the world is just phenomenal. And I really hope that competitions like this continue to help give, voices to young scientists to showcase their work and help launch them onto the world stage. Because as we've seen today, there is just incredible research being done by these people that really needs to be heard and seen. So congratulations to all my fellow competitors. You've all been amazing.
Nicely said. Well, we'll be getting one of those trophies all the way to you down under. Congratulations, Samantha. Thanks for being up as well to chat to us. That was awesome.
Thank you.
Bye. Oh, wowzers. Right. It's now time to reveal who the judges decided. Oh, no, it's not. We're going to reveal who the judges decided to be this year's FameLab International winner very soon. First, I want to do the results of your vote because I have been sent who that is. So hang on 1 second. Need to pull that up. All right. Okay. So let me make sure I can see all of you. Let's bring up the Zoom screen. This is really exciting. I have been sent the name of the person that you watching voted for as the FameLab International 2021 audience vote winner. And it is Sirawit from Thailand. Hello, hello. What? Join me. Talk.
Hello. Wow. This is fantastic. You know, Greg, this is like 1 a.m. in the morning in Thailand. I feel so flattered with lots of love and support. So thank you, everyone. You are the best.
Oh, there was definitely a lot of support in the live chat. I didn't realize it was 1 in the morning. Well, congratulations.
Talking to my Thai supporters, there seems to be like lots of people commenting like that. ขอบคุณนะครับขอบคุณนะครับทุกคนนะครับที่เข้ามาโหวตกันนะเห็นไหมว่าเนี่ยทุกคนเก่งมากเลยตอนนี้ทุกคนทำให้ไทยแลนด์ได้รับอันนี้คือรางวัลโหวตขวัญใจมหาชนจนได้ครับแล้วรอลุ้นกันนะครับว่าไทยแลนด์จะได้รางวัลที่เท่าไหร่นะครับเพราะประกาศไปแล้ว2แล้วก็ขวัญใจมหาชนเหลืออีก1รางวัลคือรางวัลชนะเลิศแล้วเจอกัน Thank you very much guys Thank you
it was great to see your you've got that
yeah
we were talking about that background earlier.
This is just like a translucent wall. It's just like glass there, and then I place, the laser pointers there, and some of the, the projectors behind me. So to create something like we are living in a universe.
Well, you're also talking about crystallography, weren't you? Tell us a bit more about your research.
So my research is about the prediction of the protein structure, which doesn't just only limit to the coronavirus. Well, actually, my project was created based on the study of human proteins. So basically, we will try to look at what kind of mutations lead to disease-causing, mutations. What kind of mutation is just benign or just okay, or the term polymorphism. So we are trying to differentiate the differences between the mutations that we found among those groups. And we found that some of the disease-causing mutations likely to cause some damages on the protein structures, and we tried to make a website application so that other researchers can use that to predict the protein structure as well. And due to the COVID-19, I tried to adapt that concept to apply that to study the mutation of the coronavirus, because there's so many mutations worldwide, and a lot of mutations didn't get studied as well, especially a lot of mutations in Thailand. So we are wondering whether those mutations are just okay, or whether we should be worried about them. And so far, lucky for us that we don't see any harmful just yet.
That's good news. And I hope that you're going to be continuing your science communication adventure, especially within Thailand, where there's a lot of support.
Absolutely. I'm going to continue being a science communicator. Greg, the first time I joined FameLab, I didn't think of winning at all. I was just thinking that I wanted to prove myself as a good science communicator. I didn't think about going to an international round. I just think about just do my best. And this is what I've come so far. This is just beyond phenomenal.
Well, huge congratulations. You are this year's audience favorite. Congratulations, Sirawit.
Thank you.
Bye. Amazing, which means it's now time for the final announcement. It's time for the big one, right? It's time to find out who the judges have crowned FameLab International Champion, and Rogério is going to announce this. Press the right button, Greg. Here we go.
Hello, it's really my pleasure to announce the big winner. And this is a talk that filled this with the height rank, these 3 C's, the content, clarity, and charisma. It was really good content. It was a really good piece of science, and it was presented in the real clear way and with charisma. And the winner is Alex from Netherlands.
I'm going to bring you straight up because I'm loving that response right now. Make some noise so we can focus on you.
I don't know what noise to make, but
Huge congratulations. How are you feeling?
I'm shocked. My pulse is 142, which is pretty high. I'm really excited. I'm shocked and super, I don't know, happy.
It was a absolutely fascinating and fabulous talk. The way you kind of hooked us with, that you work in a lab with leftover human bits, and then with the chess as a a nice bit of imagery. How long does it take to come up with an analogy and to work it through like that?
That one took actually only about 30 seconds because I was really into the Queen's Gambit when it came out, and I was like, this is a perfect analogy for the immune system, which, I mean, maybe shows you that I think about work a bit much for my own good, but it's two passions bridging together. Three maybe: Netflix, chess, and the immune system.
Amazing. I bet it took quite a long time to choreograph it, though. You know, as someone who also gives talks, especially using props, that's a tricky thing to do, especially live. You know, live when it was recorded.
Yeah, I practiced it loads. I was lucky enough to have a practice session on Zoom with a mentor, Barry Fitzgerald, the superhero scientist from the Netherlands. So that helped a lot. And yeah, the worst part was that my hands were shaking so much during the pitch that I couldn't actually flick the king at the end. I was like, that's it, it's over. Checkmate is catchy if you actually throw the king off, but not so much if your hands are going like this. But
well, you did an amazing job. And again, same question. How's this whole experience of FameLab been for you from kind of right at the start, you know, the rounds in Netherlands, right through to the international final?
It's been fantastic. I think especially in a year like this, where it's quite easy to feel isolated, having this sort of community going through FameLab all the way through from, well, the Amsterdam UMC where I'm from, the Netherlands, and here. It's been amazing to meet all of these amazing people who have a shared passion and are really good at what they do. It's really been an honor to be a part of this.
Well, you're now part of the FameLab final, which, you know, has grown and grown each year, and it's just such a wonderful bunch of people. So you're now part of that. What's next? What's next for you?
Good question. Finishing my PhD is kind of a top on the list. I also have a blog that I'm consistently working on since my master, so that will continue through. And I guess I'll kind of see where life takes me. But science communication will definitely be a part of it. It's my favorite thing to do.
It absolutely needs to be. Well, huge congratulations. Loads of congratulations coming in on the live chat as well for you from Jessica and Leo and Darby and Scott and Nancy and all sorts of people from all around the world. So huge congratulations. Best of luck with the rest of the PhD. And what's the name of the blog?
Microbial Mondays.
There it is. This is going to be the winner 2021. That is going to be coming over to you as well. So I'll pop that in the post for you tomorrow. Huge congratulations again. Thank you, Alex. Nice one.
Thank you so much.
Bye. Hang on. Apparently I was muted for the final link, which is so classic, but at least it was for the final link. So I need to do the thank yous again, because it's very important that everyone gets thanked. I'm glad it was the last link that I was muted for. Let's say a huge thank you. Massive well done to all of the finalists and to everyone who took part in FameLab, because you were all absolutely brilliant. But here are some more thank yous to our judges, to Tara, Rogério and Julia, to the semi-final hosts, Eman and Kareem. Thank you. You both did an awesome job. To the whole team at the British Council and the FameLab team at Cheltenham Festivals, especially Elspeth. Thank you also to Malcolm Love and to alumni Adriana, Veli and Roshni for the masterclasses. To the production team at Still Moving Media for the edits. My company is Science Media Studio, who have been handling this stuff here in the spare room. And that is it. My name is Greg Foote. Thank you so much for joining me tonight. Let's try that one last time. Goodbye.